So we left off last time talking about the ability of neurons to produce electrical signals, right? We talked about action potentials, we talked about graded potentials, and we've started talking about synapses, but now we're going to really break it down and get into the detail of it. So a synapse is any meeting point between a neuron and any other cell. Now, most of the time, the other cell is also a neuron, but the other cell can also be a variety of other things. So anytime a neuron is controlling an effector, so like muscles and glands, then the effector cell will be on the other side of that synapse, like the neuromuscular junction that we learned about before. And neurons can also be receiving from sensory cells that aren't neurons. So we'll be able to hit on that a little bit, hopefully on Wednesday. But basically, anytime a neuron communicates with another cell, it is a synapse, that point of contact. For our purposes, we're going to assume this is a neuron to neuron synapse because it makes it easier for me to talk about it. But just keeping that in mind, that it can be a different cell. So in order to not get confused when we talk about synapses, we talk about a presynaptic cell or a presynaptic neuron as the cell sending information. So this is the cell whose axon is terminating on another cell. And then the postsynaptic neuron is the one that's receiving the signal. So when we learned about neuromuscular junctions, the skeletal muscle cell was the postsynaptic cell, and the motor neuron was the presynaptic cell, okay? So that's, what do I say, like that makes sense because these synapses are going to be one way, right? We're always going to have one neuron sending information. We're going to have neurotransmitters crossing the synaptic cleft, and then the signal goes into the postsynaptic cell. Now, there are two kinds of synapses possible. There are chemical synapses, which use the setup that we've learned about, and neurotransmitters, excuse me. And then there are electrical synapses. So electrical synapses are where we have cell-to-cell connections, typically with gap junctions. And those do exist in the nervous system. There are neurons that are electrically connected, but they're much less common. And so we're not going to really deal with them here. You will learn about electrical synapses when you get to the cardiovascular system, because the best examples of electrical synapses are between heart muscle cells. So you'll deal with it at that point. They do exist here, but we're not going to really tackle that. So we're going to focus on chemical synapses. And this is set up the way that we've already learned, because the neuromuscular junction is an example of a chemical synapse. So this is where we have a presynaptic neuron releasing neurotransmitter across the synaptic cleft in response to, their action potentials coming down the axon. And the neurotransmitter is what stimulates the postsynaptic neuron. So because we have a chemical that's diffusing across a gap, this is always going to be a one-way signal. And that's kind of important to note, that these signals do not reverse direction ever. The neurotransmitter is always going to be released when the axon terminal is stimulated by a change in electrical potential. And so there are voltage-gated calcium channels at the synaptic terminal that are activated when an action potential comes down the axon. So we'll have an action potential coming down. It'll activate voltage-gated calcium channels. Calcium will flow into the axon terminal, and that stimulates exocytosis. So it tells the end of the axon to release neurotransmitter from stored vesicles. And then the neurotransmitter diffuses across the cleft, binds to whatever receptors are on the other side and stimulates some sort of reaction on the postsynaptic neuron, which, as we talked about with graded potentials, could be a change in charge so that it's more positive, but it could also potentially be a change in charge so that it's more negative. Obviously, the signal has to end at some point, so we do that by removing the neurotransmitter. So basically, once the presynaptic neuron is done releasing neurotransmitter, then whatever ongoing mechanism of removal is occurring will catch up and get it all the way out. Once there's no neurotransmitter, there won't be any binding to the receptors, and the signal stops. So there's 3 ways for neurotransmitter to be removed, and the mechanism used at a particular synapse depends on the neurotransmitter. So some of them simply diffuse away. Some of them are actually taken back up into the presynaptic neuron. So we call it reuptake. And this is basically recycling because they'll take it back up and they'll actually be able to reuse it again. And then some of them have enzymes in the synapse that break it down. And we saw this at the neuromuscular junction because acetylcholine is removed by enzymatic degradation. So either way, the result is no more neurotransmitter binding to the receptors, stops the chemical signal. So that being said, there are different types of neurotransmitters and different receptors for those neurotransmitters. We can divide up all the neurotransmitters into 4 groups based on the chemical structure. And we'll go through them one at a time in this order. So this is just the overview list. So we'll start with the one that we're familiar with, which is acetylcholine. So we call it the cholinergic system. It's every single receptor that uses acetylcholine at every single synapse. Acetylcholine is the only neurotransmitter in this group, but we treat it like it's a whole system. So there's 2 types of receptors. There's the nicotinic receptors and the muscarinic receptors. And their names are based on toxins that bind to the receptors because this is how a lot of this stuff was investigated originally. So there's a mushroom toxin called muscarine that can bind to these guys but doesn't bind to the other ones. And then nicotine binds to these guys but not to the other ones. So we know that there's 2 distinct receptors that will bind acetylcholine. And we know that both the use of acetylcholine in both of these receptor types are very widely distributed. So this is a very common system used in the nervous system. We see it a lot for our motor output. So the neuromuscular junctions we already learned about, as well as pretty well distributed throughout the autonomic nervous system. So remember, this is all the visceral, peripheral nervous system stuff, and we'll touch on the details of that later on. So I can't remember if I showed you this already or not, but here's just another neuromuscular junction using acetylcholine. Which, remember, the receptors are also ligand-gated channels, so when it binds, we get movement of ions. Oh, there we go. And then you don't need to know any of the details on this slide that aren't present in other places, but just trying to show you acetylcholine is very widespread and does a lot of things by way of nervous system control. So the autonomic nervous system controls a lot of stuff, including secretions from glands, smooth muscle control in a lot of places, And this is showing it doing other things as well. So just trying to find some good pictures. The second group is the amino acid group. And it's called that because amino acids are neurotransmitters. So not every amino acid is also an not every amino acid is also a neurotransmitter. However, there are several of them. We're not going to worry about the details. I gave you a couple examples, but you do not need to know them. Each amino acid has its own receptors, and there's no interaction between them. So what that means is that we don't see necessarily any connection between, you know, specific amino acids and their receptors. So there's no cross binding, there's no cross usage. They're all discrete, separate systems, just like acetylcholine is its very own system. Now, unlike acetylcholine, which is eliminated by enzymatic degradation, amino acids are typically eliminated by reuptake. So recycling is the removal system of choice, which means that they get to be reused as well, which is nice and efficient. So just a couple of examples that, again, you don't need to know. So glutamate is one of our 20 amino acids that make proteins, and it has its own system. And then GABA is a glutamate derivative, so it's been slightly altered. And this guy is super common in the central nervous system, and we actually target its receptors with a variety of drugs for all sorts of different things. So just to kind of give you some examples of the more prevalent ones. Okay, so amino acids can act as neurotransmitters. And here's a picture of GABA being used. So this is just showing you that we're altering the glutamate, making it into GABA, using it as a neurotransmitter, binding, and then the recycling is can either be by the neuron or actually by associated cells, but again, you don't need to know that. All right, so amino acids. And the other two groups are actually also related to amino acids, but altered. So biogenic amines are amino acids that have been modified so their carboxyl group has been removed. So that makes them not amino acids anymore, but their amino group, their nitrogen containing group is present. So we call them amines. It's a chemistry thing. So basically you can think about them as altered amino acids. And this is a super common, super important group of neurotransmitters. So for example, the amino acid tryptophan is altered into the neurotransmitter serotonin. Serotonin is a super important neurotransmitter. It's involved in a lot of things, especially involving like mood. So we see a lot of drugs targeting receptors in this system for things like depression and anxiety. And then another example is the amino acid tyrosine, which is made into dopamine, norepinephrine, and epinephrine. And again, you do not need to know these examples, okay? But these are, again, really important, really common neurotransmitters, and we will touch a little bit on their effects when we get to the autonomic nervous system, because that's where we see norepinephrine and epinephrine. Okay, so again, taking amino acids, modifying them, and that gives us a different group, each with their own system. So this is what happened when I looked for images of serotonin. I got some sort of weird thing. So the last group is neuropeptides. So a peptide is just a chain of amino acids, and they're neuropeptides because they're acting like neurotransmitters. So now we have strings of amino acids, and the length varies. In fact, the examples I gave you were just because they're kind of the range of the length, which again, you don't need to know the examples. The weird thing about these guys is that they're not, they're usually released along with another neurotransmitter at the synapse. And the other thing is that a lot of these guys can be released into the blood and act as hormones as well. So these are just a couple of examples just showing you the range of amino acids. So like this guy, mesencaphalin, is 5 amino acids, and beta endorphin is 31. So this guy is, when we talk about releasing endorphins, because you like exercised or something, this is one of those endorphins. Like it's the feel-good type of chemical, and it's the reason why opiates work on us, because we do make these natural ones or endogenous opiates. That's why the drugs we take are able to bind, because we already have receptors for it. So that's neuropeptides, just strings of amino acids. So basically everything's amino acid based, and then there's acetylcholine. So responses to neurotransmitters. So there's two options for responding to neurotransmitters, and this is where we talk about, this is how we talk about graded potentials, because we know that what happens to the postsynaptic neuron is going to be a graded potential. That's how they respond to synaptic stimulation. So that means that we have two responses to neurotransmitters binding, and they're both going to be in the form of graded potentials. So we can have depolarization, which means becoming more positive, and we can have hyperpolarization, which means becoming more negative. Now, which one occurs and how it happens depends on the receptor. So the big thing about all of this is I can talk about neurotransmitters as much as I want to, but it's actually the receptors that determine the effect of the synapse, not the neurotransmitter. Now, most neurotransmitters have the same effect at different places because the receptors all do the same or very similar things, but it's still the receptor that's actually doing the changing of the postsynaptic neuron. So let's take a look. We have two basic types of receptors. So there's the type we've seen before, which is actually classified as ionotropic. So basically it's a ligand-gated ion channel. The neurotransmitter binds, it opens the channel, ions move through either into or out of the cell, it changes the membrane potential, okay? So ionotropic. That's how the acetylcholine receptors work. The kind we haven't seen before is called metabotropic. And with metabotropic, the receptor for the neurotransmitter isn't an ion channel. Instead, it's just a transmembrane protein that, when bound to its neurotransmitter, is going to trigger a whole set of reactions inside of the cell. And we're not going to get into how that works because we don't have time for that. But basically, most of the time, the end result is still the same, which is to say that we're either going to increase or decrease the charge across the membrane by letting ions move. It's just that it takes a little longer to get there. Sometimes it's a little bit different because these guys can also do things like change the way that the cell is expressing proteins and stuff like that. But we'll concentrate on the way that they're both changing the membrane potential. So ionotropic is also called direct activation because it's a one-step system where we bind neurotransmitter and ions move and we change the charge. Fairly straightforward. The metabotropics are also called indirect activation because this has to like create a signal, and the signal causes reactions, and then eventually we have ion movement. So there's pros and cons to both, and we're not going to go into that, but they're both perfectly valid systems. And this is just the same picture again in case I needed to point anything else out, and I can't think of anything. So moving on. This is just a little point, and we're not going to get too caught up on it, but basically we can also categorize synapses based on where on the postsynaptic neuron the synapse is occurring. So we've kind of talked about this and made it sound like all of our synapses are between the axon terminals and the dendrites of the postsynaptic neuron. But that's not actually true. They can be anywhere on the neuron itself. So when it's between the axon of the presynaptic and a dendrite, we call it an axodendritic synapse. When it's between the axon and the body of the cell, we call it an axosomatic, because remember, soma is the other word for the body. And sometimes we'll actually have them to the axon of the postsynaptic itself. So those are called axonic. So really, we're just giving it the name of the two things that are connected, and they all sound really weird and complicated, okay? So that's kind of just as far as we're gonna go with that. It does have consequences when you get into like signaling pathways and stuff, but again, we're not going to dig into that because we don't have time. Okay, just a little bit on electrical synapses. Again, we're not going to talk about it a bunch because you'll learn more about it when you get to the cardiac muscle physiology, but they do exist in neurons. And so just want to touch on a couple of differences between these and the chemical. So again, now instead of being joined by a chemical synapse, we have direct electrical connection with gap junctions. So we have flow of ions directly from one cell to another. The biggest difference here is that now we have a signal that can go both directions, and it's much faster than the chemical kind. So we don't have that waiting for the neurotransmitter to be released and diffusing across the synapse, and then it binds, and then it has an effect. We have an immediate movement of ions. But it's not always beneficial for a signal to go both ways, so we need to use chemical synapses when we want to control the direction of the signal. When everything is allowed to respond the same way and we just want a signal to spread really rapidly, then we use electrical synapses. So that's why we have both. So gap junctions, nice picture from Wikipedia showing you how they can connect and that they can be open or closed. And then this is just a diagram I found when I was trying to find pictures of synapses. Turns out you can have mixed synapses that are both electrical and chemical, which is kind of weird. But, you know, there you go. All right, so that leads us to a little bit more detail about graded potentials. So what we didn't talk about before is that on the receiving end of graded potentials, when we're getting sensory information into the nervous system, there's two different kinds. And we'll touch on how that works when we get to the sensory stuff, again, hopefully on Wednesday. So basically, sometimes the sensory receptor is a neuron, and sometimes the sensory receptor is like an epithelial cell connected to a neuron. So when we have a sensory neuron acting as the receptor, and it's stimulated by whatever it's sensitive to, pain, temperature changes, vibration, whatever it's sensitive to will cause graded potentials in the membrane. So we call those generator potentials. And if the generator potentials are strong enough, then it'll eventually fire off an action potential. with sensory cells that are connected to sensory neurons, we call them receptor potentials. And instead of leading to an action potential, because it's a sensory cell, it can't have one. So instead, it actually has a chemical synapse, and it releases neurotransmitter onto the receiving sensory neuron. The more graded potentials it has, the more neurotransmitter gets released, and this, if strong enough, will usually result in an action potential from the sensory neuron. So it's just like one extra step when we have additional sensory cells. So little diagram on generator potentials over here. So sensory stimulation, graded potentials, potentially leading to action potentials. And then with receptor potentials, graded potentials here lead to neurotransmitter release, and then this can eventually lead to action potentials. Okay, so an extra step. And then it'll make more sense when we hit on sensory stuff, when we get to peripheral nervous system. So that leads me to think that I thought we talked about already, but we didn't, which is fine. It's totally fine, which is our postsynaptic potential options. So we can get two options of graded potentials on the receiving end of a synapse. We can either decrease the charge and have the membrane become less negative, or we can increase the charge and have the membrane become more negative. So we call these postsynaptic potentials, and we abbreviate that to PSPs. When we depolarize the membrane, we call them excitatory postsynaptic potentials, or EPSPs, because we're exciting the membrane, making it less negative, bringing it closer to threshold, and potentially getting an action potential. But if we make the membrane more negative, we call it hyperpolarizing. And we call those inhibitory postsynaptic potentials, or IPSPs, because now we're making it less likely that the neuron is going to have an action potential. We're inhibiting the action potential from happening by becoming more negative. Tell me that I have it. That's going to do. Okay, so the EPSPs get us closer to threshold, and IPSPs take us farther away from threshold. And these guys, if they're big enough and they actually take us to threshold, then we actually get the whole action potential. So that takes us to the last point about action potentials, which is how, or graded potentials, sorry, which is how do we get to action potentials? So we talked about action potentials initially, and we know that they're all or none, and we know that once they start, they go all the way down the axon. And we know that graded potentials vary based on the strength of the stimulus. We know that they can depolarize or hyperpolarize the membrane. So if we get enough graded potentials depolarizing the postsynaptic neuron so that they depolarize the membrane at the axon hillock to threshold, then we get an action potential getting sent out. And usually this takes the accumulated depolarizations of multiple inputs, because neurons have multiple synapses. Usually there's a bunch of them, and if they get signals from many of them at the same time, it's the total effect that determines whether or not they get to threshold. So we call that total effect summation, and that's ultimately what determines whether or not a neuron fires at any given time. We can have summation in a couple different ways, just like we can when we're looking at muscle contractions. So spatial summation is when a bunch of presynaptic neurons fire and they depolarize the postsynaptic neuron to threshold by all basically firing at once. So if we have a bunch of depolarizing or excitatory graded potentials, we can get to threshold. Temporal summation is when a single very determined presynaptic neuron is firing over and over again strongly enough to depolarize to threshold. So spatial summation, because across the space of the neuron we have a bunch of them all at once. Temporal summation, because if you hit it fast enough from the same source, you can get to the same thing. So this is, it's easier to demonstrate spatial summation. So this is showing you a bunch of presynaptic neurons that are firing. All these green guys are depolarizing, and then the pink guys are hyperpolarizing. If the total of depolarization gets this point to threshold, though, we get an action potential. So the axon hillock is the thing we need to get depolarized. Okay, anybody have any questions on that chapter? All right, let me switch. Okay, so we're gonna at least talk about the brain today. I'm supposed to talk about the whole central nervous system, and I'm guessing we're gonna run out of time, but that's okay. The spinal cord is not as complicated, and we'll just roll it over with the peripheral nervous system on Wednesday. So we're gonna start by very, very briefly looking at how the brain develops initially. And I only do this because I think it helps you to understand the adult structures, not because I think it's critical that you understand the embryology of it, which is interesting, but we don't have time for that. And then we're going to go through how we organize the central nervous system, the major structures, and the functions of those structures, and then we'll see where we're at. So I'm guessing that this is all going to be Wednesday, because this part takes forever. I cut it back as much as I could, but either I'm bad at that or I'm probably just really bad at it, but it all feels important. Okay, so like I said, we're going to start by very briefly looking at how all this develops. Even though we haven't really talked about any embryology before, I think you'll be fine. So at the very beginning of development, there's the embryo just kind of starts as a couple of layers of cells, okay? So there's 3 layers of cells, and one of those layers is the outermost. It's called the ectoderm. And some of the cells in the ectoderm become the nervous system. Some of them also become the integumentary system. So the very outermost layer of cells that we start with becomes both something very central and something covering the surface, which is super weird, but that's the way it works. Now, the part of the ectoderm that's going to become the nervous system actually ends up sinking below the surface. And it does that by specializing and forming what's called a neural plate that then sinks down below the surface. So it does it in a fashion that we call a neural groove. So literally it sinks like this. And then this part is also special for nervous system stuff. So those are the neural folds. And then it keeps going. The folds actually come together and we get this neural tube and a neural crest. So those two things, and this is on cross section. So what we see in the whole embryo is kind of this business, a tube running down the length. when this happens, we end up forming sort of a head direction and a tail direction, and the length of this then becomes the nervous system. And you can see it's kind of already set up like a spine. So here's the fancier version of that. So this neural tube then starts to fold because, you know, our spinal cord is basically a cord, but our brain isn't. Our brain is a lot of things. So this thing starts folding like a you know, one of those balloons that you make little dogs out of. And so as it folds, the structures, we call them vesicles. So we start with 3. And the nice thing is that they correspond to the 3 major regions of the brain. And that's the other reason I'm pointing this out. So Prosencephalon means forebrain, Mesencephalon means midbrain, and Rhombencephalon, we're going to roughly translate to hindbrain, even though that's not really what it means. And encephalon, you know, it means brain. So don't get too hung up on how complicated those words seem, because it just means this stuff. So that starts the process of ending up with these separate structures. And then there's going to be some more folding, okay? So here's our first series of vesicles. So forebrain, midbrain, and hindbrain. The rest of this is going to be spinal cord. And you can see this starts very early. This is within a month of fertilization. And then we get secondary vesicle formation. So the 3 primary ones do some extra folding. So the forebrain becomes 2 separate things. So the telencephalon becomes the cerebrum. So that's we'll talk about it. The Diencephalon becomes these guys, the thalamus and the hypothalamus. The Mesencephalon is already as specialized as it needs to be for further development. And then the hindbrain does something similar and splits into 2. The Mesencephalon becomes the pons and the cerebellum. And then the Myelencephalon becomes the medulla oblongata. And I promise that every single one of these words that I just introduced you to, I will use again very shortly, and you'll see which part is which, okay? So that's kind of the basics of brain formation. So when all is said and done, all of this ends up being your brain, and then everything else of the neural tube is your spinal cord. And the other thing I want to point out here is the beginning of eye formation comes right off of your brain, which I think is pretty cool. All right, so that's kind of your summary of how all that comes to pass. And really all you need to know is on this chart or diagram, which is a single diagram that I cut in 2 so that it would fit better on the slide. All right, so the rest of the neural tube becomes a spinal cord, as I said. Doesn't need to do any convoluting because the spinal cord is already, you know, it is a tube, so it's all good. The one thing that we do see is that the dorsal or posterior aspect devotes itself to becoming sensory related neurons and stuff, and then the ventral or anterior aspect becomes motor neurons and stuff. And so we'll see how that plays out when we get to the spinal cord on Wednesday. This was the best I could find of another picture of that. So basically the neural tube, we already saw this happen, but now we're looking at this part of it. And so basically this will become sensory and this will all become motor. So very well divided and well organized, even at the beginning of its formation. All right, so now we're going to take all that vesicle talk and we're going to see how it corresponds to the regions of the brain in adults. We're also going to hopefully talk about the protections of the brain. Actually, I think we are. I think that's first. Anyway, we're going to start with this though, because I think this makes it more interesting. So there's a lot of like myths and false perceptions about how our brains work. So the one that loves to travel around all the time and has even become the main plot point of movies and stuff is that humans only use 10% of their brain. And so there's this idea that you could do something to use more of it and then you'd be like some super genius or something. But that's not actually true. We use all of our brain, and hopefully I'll demonstrate that by telling you what all the different parts are doing, and then you can see that we're really using all of it. Another thing that hopefully we'll have time to get to is this idea that what you remember is accurate, that what you remember is like video recording of something that actually happened. And it turns out that's not true. When we remember things, we recreate the memories, and they change every time we remember them. So unfortunately, we can't trust the factual nature of a lot of our memories, which is kind of weird. Another one is that our senses tell us exactly how the world is. So our senses are accurately portraying what's actually happening. And we won't get to this right away, but when we talk about sensory perception, we'll find out that's not true either, because our brains process all of our sensory information before we become aware of it. And so there's a lot of stuff it leaves out from our conscious awareness. And then another one that drives me nuts is this idea of right brain and left brain. This idea that like people that are right-brained are really creative and artistic and whatever, and left-brain people are really like analytical and mathematic and stuff like that. And that's not how it works either. All of us use both sides of our brain, and although there are a couple of things that are done only on the right and only on the left, it doesn't work that way. Nobody's right-brained or left-brained. Well, there are very, very rarely people who are literally missing half their brain. So they are, but that's obviously special circumstances. All right, so we can divide up the brain into 4 regions, okay? So we've got the cerebral hemispheres, that's the Telencephalon. We've got the Diencephalon. We just keep calling it that because it works best. The brain stem, which is the, well, these guys all together are formed from the hindbrain, and it doesn't really quite correspond evenly. So these guys are all hindbrain structures. And then there's related supporting structures, the meninges and the vesicles. Don't confuse these vesicles with the embryonic ones, okay? They're totally different. These guys are fluid-filled structures containing CSF, and we'll talk about that in a second. So, basic brain, you've probably seen this before. So here's your cerebrums or cerebral hemispheres. The Diencephalon is in here, basically right here. And then your brain stem is down here, and your cerebellum is here. So that's how we're gonna move through it. We're gonna start up top and work our way down and see that all of these parts have different roles to play in our nervous system function. Because otherwise I end up forgetting, I like to just start with the blood supply. So remember that nervous tissue is very metabolically active. So really, really needs a constant supply of oxygen and glucose. And so that means that the blood supply is super important. The internal carotid arteries are the major blood vessels that supply blood to the brain. But we also do get some from the vertebral arteries. So basically, you know, when you can feel your pulse, that's your carotids. So that's your main blood supply. And then there's vertebrals, which kind of run up your vertebrae. So hopefully you guys remember that those cervical vertebrae have those transverse foramina. Right? Those are the arteries that run through there. And so there's these carotid canals that I didn't make you learn in the base of the skull, and then the vertebrals run up through the foramen magnum. with the spinal cord. And then everything merges kind of at the base of the brain to form this guy called the basilar artery. And there's this cool thing in there called the circle of Willis. It's basically a roundabout for your brain. And the idea there is that if any of your arteries get blocked, which we know happens, and that's what can cause like a, like a stroke. If that does happen, then the blood keeps going around the circle and can get to those areas another way. So we have a lot of redundancy built into our blood supply to try to prevent damage if blood vessels get blocked. So they call this the circle of Willis. There's probably a modern name for it too, but I don't remember what it is. And where'd they go? Oh, so then these are the carotids coming in from, from your neck and up through the base of your skull. And then these are the vertebral arteries coming in through the foramen magnum. And then this is all sitting sort of, so if you look at that brain diagrammy thing again, that's sitting about there. And then we have these super big veins that drain all the blood. You imagine there's a lot of blood draining because there's a lot of blood going in. Veins that are super duper big are called sinuses. And the big guys are actually inside of the meninges, which are the connective tissues protecting the brain. So we call them dural sinuses. And there's a bunch of them. You don't need to know these. But basically everything drains out into your jugular veins, and your jugular veins run along with your carotid arteries. So the carotid arteries are sending blood to your head. The jugular veins are draining it out of your head and heading back to your heart. So it's kind of like this. So there's a lot of veins like kind of surrounding your brain structures to drain everything out, and they like to like merge together and stuff, and then they end up going out down your neck. All right, so other thing before we actually get to the brain is stuff that protects your brain. So I like to do it like this again, A, because I don't want to forget, and B, because I like to go from the outside in. I don't know why, it just makes more sense. So there's actually several different layers of protection between the central nervous system and everything else. It's super important that your neurons are kept completely separate from the external environment because they need that very precise chemical environment of ions in order to function. So there's basically 4 levels of protection. There's 3 levels of physical protection. So the bones of your skull, the meninges, and then we've also got the cerebrospinal fluid. I'm going to talk about each one of these in turn, I promise. And then there's also the chemical barrier of the blood-brain barrier. So we'll go through each of these, except for the bones of the skull, because we talked about that already. So bones of the skull, we know we have them, and we know what they're for, right? Flat bones, protect your brain. Okay, so deep to your bones is the meninges. The meninges are actually a 3-layer connective tissue barrier protecting the central nervous system. So they're kind of like the periosteum and like the epimysium of muscles and stuff, but way more intense. So they cover and protect both the brain and the spinal cord. They also protect the blood vessels and venous sinuses, which is why I showed you that drainage of blood picture. They're in the dura. They help contain the cerebrospinal fluid so that it's able to actually surround the brain and the spinal cord. And then they actually also form partitions inside the skull, which divide the major parts of the brain from each other. Why? I don't know. I think it's just an organizational thing. All right, so 3 layers, right? All 3 layers have a name that's got a descriptor and then mater. Mater means mother. So because they are all encasing and protecting the delicate brain, the meninges are the mother of the brain. So the outermost layer is called the dura mater. Something that's durable is very tough and sturdy. So this is 2 layers of very tough, dense, fibrous connective tissue. There's actually a layer attached to the inside of the skull bones called the periosteal layer. It's, I believe, literally also the periosteum of that bone. And then there's a meningeal layer that directly covers the brain and spinal cord. Now, in most places, both of these layers are fused, except for where they part to allow the dural venous sinuses to go through, and the dural septa, which are the parts of the meninges that subdivide the brain into different sections. That's just the meningeal layer. So I'll show you a picture. So these are the dural septa. So basically, the dura mater is stuck to the inside of the skull bones, except for where it divides the brain. So the cerebrum has a left and a right hemisphere, and it goes in between them. And then there's a division between the cerebrum and the cerebellum right here. Cerebellum down here, cerebrum up here. So there's this guy, and then there's hemispheres in the cerebellum as well. So the falx cerebri separates the left and right sides of the cerebral hemisphere. That's this guy. The falx cerebelli divides the hemispheres of the cerebellum, and then the tentorium cerebelli separates the cerebellum from the cerebrum. And that one I just remember because it's like a tent over the cerebellum. So these are indwellings of the dura that I think it just provides a little extra protection, and also because you can see that the sinuses are running down here too, it also provides this stable pathway for these dural sinuses. All right, middle layer is called the arachnoid mater or spider mother because it's very loose, spacious connective tissue, and it kind of looks like a spider web. In between the dura and the arachnoid is a fluid-filled space of, it's called the subdural space. This is also the space that people bleed into when they have head trauma. So you get those subdural hematomas because there's space there. And then between the arachnoid and the pia, there's CSF as well. And there's big blood vessels in here. And then we'll talk more about these arachnoid granulations in a minute. Okay, so there's space. It's all about space with the arachnoid. And I don't really have a good picture, so I'll show you another picture in a second. The pia mater, pia means gentle, so this is the gentle mother. So this is just a very thin layer of connective tissue along with all the tiniest blood vessels that literally follows all of the contours of the outer surface of the brain. And there's a lot of contours, so it also goes in a decent ways because there's a lot of infoldings. And so this is the one that like you can't really easily separate from the brain tissue itself. It's stuck to it. And so really does do a lot to kind of hold the brain shape in place. So this guy is very similar to like the endomysium that surrounds each individual muscle fiber. It's just this very thin, delicate little connective tissue layer. So this is a picture, frontal section. So what we're seeing here is skull bone. And then we've got the dura mater here. The layers separate to go to form this falx cerebri. So this is brain here. All of this stuff is the arachnoid, and then the pia is right here. So then there's fluid in between the dura and the arachnoid, subdural space, and then there's lots of CSF in the subarachnoid space between the arachnoid mater and the pia mater. All right. So that takes us to the CSF itself. And again, if you have any questions, just throw something in the chat or turn your mic on and ask, or whatever you need to do. So cerebrospinal fluid, which from now on I will say CSF, now that I've officially defined it. Basically, its job is to suspend the central nervous system in fluid. So it's this liquid cushion that you're floating in, that your brain and spinal cord are floating in. And that serves a couple of different purposes. So first of all, it protects your brain from being damaged by its own weight. So literally, if our brains were not buoyed in fluid that they were floating in, then the weight of the brain would damage this, whatever structures were sitting on the base of your skull due to gravity. It also protects the CNS from damage from trauma. So anytime we come to like an abrupt stop or something, this helps keep the brain from like literally smashing into the skull. You know, the way we get jarred when we have to come to a hard stop in like a car or something. Prevents that from happening. Sort of important. And then it also helps provide nourishment to the brain and probably carries hormonal signals and other signals around the brain as it circulates. We're still learning more about those types of things, but we know for sure that it does this protective stuff. The CSF gets formed from plasma, so the liquid portion of blood. And that means that mostly it's water and ions, and like probably some glucose and stuff. There might be some proteins in there that, but pretty much it's just salt water of varying consistencies. So remember that the ependymal cells are responsible for forming the CSF. They do that in conjunction with what's called the choroid plexuses. So the choroid plexuses are just clusters of capillaries, so the teeniest, tiniest blood vessels. They're covered in pia mater, and then the ependymal cells are the next layer. So then the ependymal cells control what exits the capillaries and enters the CSF space. And where the CSF goes is there are ventricles, so fluid-filled spaces inside of the brain. And then there's the subarachnoid space that we've already talked about. And it circulates all the way through from the ventricles out to surround the brain and the spinal cord because of the cilia that the ependymal cells have. And then it actually gets reabsorbed by blood vessels called arachnoid granulations, which return the fluid to the blood in the dural sinuses, where it then drains back to the heart. So basically, we're constantly forming and absorbing CSF at an equal rate, just in different locations. So the sinus, or the vesicles are mostly in here. I hope I have a better picture, but we have a third ventricle, we have lateral ventricles, and then we have fourth ventricle, and they're all connected. Here's the fourth ventricle here. And then there's a central canal running through the middle of the spinal cord as well. So that's all the internal CSF. And then you can see all the little arrows showing all of that subarachnoid space. So it's going all through here. And then these arachnoid granulations are actually these little places where it sticks up through the dura. and the subdural venous sinuses just directly absorb it. So it gets produced in here, circulates through the ventricles, and then out and around everything, and then gets reabsorbed up here. And they're very three-dimensional, so it's kind of hard to do, but we've got a third ventricle. We have a left and a right lateral ventricle, so these are sitting on either side of the third ventricle, and then we've got the fourth ventricle here. Really should find a 3D one. I'm pretty sure I'll be able to find you one for lab. All right, so those are the ventricles. They contain CSF. So then the last protection is this blood-brain barrier, or BBB. I don't think anybody calls it that, but it's way more fun when you're studying it. So the idea of the blood-brain barrier is that we're controlling everything that exits the blood to go to the neurons. So it's a control over what passes into the extracellular environment around the neurons, because again, we want to really tightly control that chemical environment, because otherwise electrical activity gets messed up. And also because of the really high metabolic rate, the neurons are really sensitive to toxins, so we want to make sure that there aren't any around them. So there's 3 layers forming the blood-brain barrier. So the capillary endothelium is the simple squamous epithelium that forms the walls of these tiny blood vessels, and they have way more tight junctions than normal capillaries. So normal capillaries are a little leaky. You'll learn about that when you get to the cardiovascular system eventually. And these guys are a lot less leaky than usual. Remember that all epithelial layers have a basement membrane, and this one is super thick. So we call it the basal lamina. So it's a really thick basement membrane. And then finally, and most importantly, we have the astrocyte processes. So remember, astrocytes are the glial cells of the central nervous system that support and protect neurons. And they're called astrocytes because they look like stars because they have so many processes coming off of them. Well, those processes in part sit on and surround these capillaries. They actually tell the capillary cells to make those tight junctions. They probably tell them to thicken up the basal lamina, and they directly control everything that leaves the capillaries and diffuses across into the actual central nervous system. So between the astrocytes at the blood-brain barrier and the ependymal cells controlling the CSF, we very, very tightly control everything that has access to our central nervous system neurons to, again, have a chemical protection. So here's a little diagram of that. This is a capillary. These are the endothelial cells, very thin. And then we've got these thickened, what do you call it, basal membrane. Don't worry about the parasite. He's just there. And then these are all the processes. So you can see that they pretty completely surround it. I don't know why there's neurons here. I assume they're allowed. But these guys are controlling everything that's coming out. So we call that selective permeability. So the ability to control what moves in and out of the capillary beds. And it kind of depends on all the same things that control what's able to diffuse. So all of the diffusion stuff we learned about at the beginning essentially dictates what happens here. So some things are able to move really freely because the nervous system needs them, so they have channels available. So glucose, amino acids, and some electrolytes are freely able to move across because they have all the channels that they could possibly need to do it. Some things are deliberately kept out, so all waste products, proteins, and anything else that's big, as many toxins as can be kept out, And then potassium ions are the exception to the electrolyte rule. We do not want potassium in the extracellular space because we need it intracellular for ideal electrical activity. So it's not allowed to come out of the blood into the extracellular space. Now, because all of this stuff is still lipid bilayers, there are some things that cannot be controlled. So obviously oxygen and carbon dioxide can't be controlled because they freely diffuse. They're small, they're nonpolar. And then anything that's lipid soluble also cannot be controlled because they can freely diffuse through the plasma membranes. This is not usually an issue, but occasionally there are toxins that get to use these rules and then they can freely diffuse, and that can be bad. So usually that's not a bad thing, but just something that especially like pharmacologists need to be aware of. Okay, so it's selective permeability. Now, there are some gaps in the blood-brain barrier. There are a couple of places where there is a direct connection between the blood supply and some of the nervous system. And those areas are called the circumventricular organs. They are around the third and fourth ventricles, and they are necessary so that the parts of the brain that monitor the blood are able to more accurately monitor the blood. So this is mostly at the hypothalamus, and then there's a vomiting center of the brainstem that monitors for certain blood conditions that will trigger vomiting if necessary. So this is really, really important because the hypothalamus does a lot of super important things when it comes to monitoring, but it also means that these are the vulnerable places where things can get in to the central nervous system. So stuff that would not otherwise be able to gain access can gain access at these places. All right. Any other questions? We're going to actually talk about the brain now. I am doing horribly at actually getting things done, but that's okay. Figure it out. All right. So starting with the cerebral hemispheres, and we'll go from there. So the cerebral hemispheres are described as the paired superior portions of the brain. And when people think about the brain, honestly, this is the part they're usually thinking about. And it's most of what you see when you look at the brain from the outside. We actually divide the cerebral hemispheres into 5 different lobes because each of those lobes are separated by really deep, we call them sulci. So they're very deep indentations. The lobes are organized so that there's gray matter superficially, white matter deep to it, and then there's some very deep gray matter called the basal nuclei. All of these lobes have additional surface area that fits in more neurons per their size because of the gyri and the sulci. So it's this convoluting of the brain surface. So gyri and sulci look like this. So the gyri are the little hills and the sulci are the little valleys. Some sulci are fairly shallow, and then the deeper ones are what determine where one lobe starts and the other one ends, or something like that. So this hopefully looks familiar. This is our typical brain picture, and like I said, most of what we see here is the cerebrum and the cerebral cortex. So cortex is an outer layer. So cerebral cortex actually specifically just refers to the outermost part, but often we refer to them as the same thing. So that's a deep one dividing up 2 lobes. That one is dividing 2 lobes, and that should be there. And then this dotted line is something else that we'll get to, but there is an internal lobe too. So there's 4 external lobes and there's actually a 5th internal. Now the right and left sides are divided almost completely by this longitudinal fissure. So all of this is just dura mater, and then there's only a connection down here, which we'll talk about in a second. So there's one with actual color showing you the different ones. So These lobes actually correspond to the skull bones that are over them. So this is frontal lobe, temporal lobe, parietal lobe, and occipital lobe. And then there is a deep lobe called the insula because it's insulated from the other ones, and you can't see it unless you kind of spread apart the outermost lobes. So here it is with a cutaway. The insula is here. It's basically deep to the temporal and parietal lobes. So you can see it right here. So you can see there's a lot to this. We fit a lot of brain into a relatively small space. And the cerebral cortex, of course, again, like I said, it's what you usually think about when you think about the brain. And it also does most of the stuff we think about brains doing. Give me 1 second. Must go. So the cerebral cortex is the location of our conscious mind, as in what we're aware of and what we think of as ourselves, and what we, like thinking and stuff like that. That's all done here. This is where our higher brain functions occur. So our ability to communicate, our ability to remember things, our ability to understand things, our ability to decide to do things. So where we decide to do voluntary movements, all of that is here. And some of it is very, very location specific. And some of it uses a bunch of different areas working together. So this is sort of a generalized functions of each lobe type thing. So for example, the parietal lobe is mostly involved in receiving sensations, especially general sensations. The occipital lobe is mostly about what you see, processing everything that you see. Temporal lobe is where some of our hearing stuff is involved. The frontal lobe does a lot of the really technical thinking type stuff. I have more information later. So we can divide all of that up, though, into 3 types of functional areas that correspond to the 3 functions of the nervous system. So there's motor areas, sensory areas, and association areas. So basically, sensory areas are where all the different sensations are received from the peripheral nervous system. Like everything comes into different areas. There's areas for all the general senses, there's areas for the special senses, and the interesting thing is that each hemisphere, so each side of your brain, receives all the input and controls the opposite side of the body. So your right hemisphere controls the left side of your body, and your left hemisphere controls the right side of your body and receives that sensory information too. Now, keep in mind that there's a lot more to this than we're talking about, and that there's plenty of stuff we don't even understand yet, okay? So this stuff's super complicated, still being studied, will be studied for decades to come, or who knows how long it's going to take us to understand it all, or if we're ever able to. And we're just skimming the surface. So the sensory stuff is where we process, or where we receive. The association areas are where we process everything. So this is the integrative function. And then the motor areas are where we actually sort out what we're going to do based on what we've processed. Okay, the 3 functions: sensory, integrative, motor. So this is an example of some functional areas. So all of our general senses stuff, for example, comes in right here and gets processed. And then there's a lot of association areas. So integration happens here for that information. And then when decisions are made, the motor output gets controlled here. And then this is showing you some areas for sight, for sound, and this is for speech, actually. The general sensory stuff that I just showed you, the interesting thing is that it's arranged very much the way the body is organized. And the motor output is the same way. And so when something looks like a person, we call it a homunculus. It's very creepy, actually. But basically it's because it's very much organized. So like your face is here, and then it's like, you know, your hand, arm, trunk, leg, and then for some reason your genitals are over there. So some stuff's not really organized like that. But anyway, most of it's what we call an anatomical map. So we call this the sensory homunculus. And then there's also a motor homunculus. They're very similar but slightly different because the size of the different areas corresponds to the sensitivity. So we can see that there's a lot of sensory stuff for your hands and your face, because we have a lot of, you know, we can feel a lot there. Whereas like your arm, your leg, not as sensitive. And then the motor output corresponds too. So we have a lot of fine motor control in our hands and a lot of fine motor control in our facial expressions. And so we've got a lot more muscle area to that too. So it's kind of cool. But creepy, so creepy. Sorry about that part. Okay, so although most stuff happens on both sides, because whatever's going on the left is for the right side of your body, and whatever's going on the right is for the left side of your body, there are some things that actually only are found on one side or the other. And so those areas, we refer to that as lateralization of cortical function. And this is, I think, where the myth of right brain versus left brain started. So whichever side actually has language-related areas, because language-related areas are bilateral. If it doesn't matter which side of your brain it's on, it controls all of your language stuff. Whichever side actually has it, we call that their cerebral dominance. Interestingly, 90% of people are left-sided dominant, but it's not a guarantee. The other thing to keep in mind about this is that even though we can have certain areas that are only on one side or the other, it doesn't mean that like the other side of the brain is slacking off. The corresponding areas are doing other things, and the hemispheres are in constant communication because there is a whole bunch of connections between them, even though they're mostly separated. So the connections are the job of the white matter. So everything I've said so far has been about the gray matter. So white matter, remember, is the axons of the neurons. And there's 3 different types of connections that we can have with the cerebrum. So basically, we can have axons connecting cell bodies within the same hemisphere. We call those association fibers. We have axons connecting the 2 hemispheres, which we call commissural fibers. Mostly that's in the form of what we call the corpus callosum, but we also have anterior and posterior commissures. And then we have to have the cerebral cortex in communication with the rest of the nervous system. And so all of those are projection fibers. So here's the white matter. So remember, tracts are what we call bundles of axons inside the central nervous system. So association fibers connect neurons or nuclei, because those are groups of neurons within the same hemisphere. Commissural fibers connect them between the left and the right. So this is the corpus callosum. It's the major connection between the left and the right hemisphere. And then the projection fibers connect to everything else in the brain and spinal cord and peripheral nervous system. All right, so those are the white matter tracts. So although most of the gray matter is in the cortex, so most of the nuclei or the cell bodies are in the cortex, that superficial layer that we've already talked about. There is some deeper gray matter in the cerebrum, and they're called the basal nuclei. Used to be called the basal ganglia, which was very confusing since ganglia should be in the peripheral nervous system, and so you'll still see it like that in a lot of textbooks, but it's basal nuclei because a nucleus is gray matter in the central nervous system. There's a lot of them, but they're all kind of just clustered together. You don't really need to know any of this, so don't worry about that. Just know that they are basal nuclei, and they are still very important, even though they're not in the cortex. So this is all the cortex stuff. All right, this is cortex, cortex, cortex. And here's all our white matter. And here's our basal nuclei. Okay, so literally at the base of the cerebrum, and here's your corpus callosum. Sorry, I shouldn't do all of it in the same color, but it just takes so long to change it. So the basal nuclei receive input from the entire cerebral cortex, which makes them super critical. And although there's a lot of, you know, detail to this, but if we want to summarize it, the overall thing that they seem to do is to remove incorrect and inappropriate responses in cognition and emotion. So it's like the monitor for your brain, in that it's like, oh no, don't do that. You shouldn't do that. That's probably not a good idea. And then there's also an influence on muscle movement. Movement is very, very complicated. It involves a lot of different parts of the brain. So we know that especially for initiating, stopping, and then tracking appropriate intensity of movements, basal nuclei do have a role in that as well, which kind of makes sense. It kind of goes along with, you know, making sure that all your responses are appropriate and correct. So yeah, important stuff. And I think if you want to think about it as the monitor for the cortex, that would be a good little generalization to help you remember. This is a little bit of best I could find for an image of how this works. So the basal nuclei are being referred to here. And then this is talking about different neurotransmitters being used, which you don't need to know any of this, but I just want to point out that they are all ones that we've mentioned. And so this is just showing the cortex is sending information to the basal nuclei. All of the information it receives is coming up through the thalamus, which we're going to talk about next, except that we're really out of time. So what we'll be doing is talking about it on Wednesday. So we'll start with the thalamus next, because we're going to tackle the midbrain. All right. Does anybody have any questions about anything before I go? No. So again, for those of you that weren't here at the beginning, uploading the tests into Canvas is taking me forever. So your lab practical will be available this afternoon, but it's still going to take me a couple of hours to actually get it all in there. So you'll have it, it'll be available all through this evening, as well as all of tomorrow. If, you know, if your only time today was like right now, it'll be available all tomorrow as well. It's just a really, really slow, tedious process to get it in. So hopefully I'll figure out how to streamline it as I keep doing this. And luckily we don't have too many more of these. The practicals definitely take longer than the lecture tests. So again, keep emailing me if you're having problems. Trying my best to tackle everything, and we will have our nervous system pre-lab stuff starting on Wednesday. I know none of you are thinking about that right now, but that's what we'll be tackling next after we finish up on central nervous system on Wednesday, and hopefully peripheral nervous system too. Okay, so hope everybody has a good beginning of their week, and I will talk to you guys on Wednesday, and I will send any announcements if anything changes. All right?
All right.
Any questions?